Okay. Hope that'll stop shaking. Hi, biology. Welcome to Meiosis, Making Babies with Sesame Street characters, if you are the father. So this will walk you through like how to separate your chromosomes and get them into your chart if you are male. Currently, this is my male character. He has 2 of chromosome number 1, 2 of chromosome number 2, an X chromosome that he got from his mother, and the Y chromosome that he got from his father. His overall genotype is little e, big N, little n, R and O, an X that is fuzzy, and a Y. So altogether he has endo eyes that are in his face. He's magenta, which I don't have magenta, but I have pink. The eyes are in the face, not on top popping out. The nose is pink because we got a dominant pink nose, and he's fuzzy all over, not just hair on the top of the head, smooth body with clothes. So that is this father's current genotype, and we're going to see how he makes sperm gametes to donate various combinations of these traits to his children. Okay, so first step is we're going to take one parent cell. It is the parent cell that is here. These are the letters that I have left over from my mom, but you would fill in your homologous pair that you have for chromosome 1, and that I have 2 little e's, a big N, and a little n. To the homologous pair for chromosome number 2, that I have an O and an R, and then 1 X and 1 Y. These technically aren't a homologous pair. You can see they're like very different sizes, and they don't have the same genes that line up like the others do, but this pair goes together as the sex chromosomes. So this information goes in your first circle. Try your best when drawing your chromosomes. You can do something like this, and then label like there's my E and like there's my N, whatever they are. You can just do kind of like a stick figure, and then an E and an N. Like just make sure that you've got your letters there. How these look, I don't personally care, but a really big, fat, fuzzy one make it hard to get all 6 of them drawn into the cell. Okay, so before we can split our cell, we actually have to replicate all of this DNA. We only have like 1, 2 copies of everything. We need to get 4 cells in the end, so we have to double down on all of this DNA at the beginning. So this actually happens in S phase or synthesis, which is even before meiosis starts. This is our germline. This is testes for a male, but this is the very beginning steps of preparing to make sperm. So we double everything. Double this one, and notice the exact same letter gets written if we're doubling the DNA, right? It's making an exact replica. There could be mistakes during this process. Those are genetic mutations. And that kind of creates possibly some new alleles, but we're not going to include that today. All right, so everything is doubled. They are still connected. I'm not going to use the masking tape right now because I forgot to bring it here, so I'm just going to keep going. But this still counts as 6 chromosomes. 1, 2, 3, 4, 5, 6. Even before, it was still 6, unreplicated. This is 6 that are replicated. That's the difference in counting. We have 1, 2, 3 homologous pairs. This is the pair for chromosome 1, the pair for chromosome 2, and the pair of sex chromosomes. So 6 chromosomes total, 3 homologous pairs. Okay, we're gonna move into prophase 1. I got my dice. Oh, sorry. Your replicated DNA gets drawn in right here, next one. So you could draw them as little X's or just 2 L's kind of right next to each other, but they are still all connected. I'm just not using the masking tape to speed myself up right now. So we need prophase 1. There we go. So in prophase 1, homologous pairs are going to join up and cross over. Right, they're going to kind of shuffle and trade some of their genes. Switching. The X and the Y will not do this. Remember, they don't have the exact same genes to trade. The mom, if she has 2 X's, right, This would have been our mother's chromosomes, here and here. Okay, replicated with big F, replicated with little s. The two X's could cross over and trade because they are identical in the genes that they carry, but the X and the Y don't do that because they carry completely different genes. So this kind of gets passed on as is, and this gets passed on as is. So now I do need some tape to refuse some mixed up chromosomes. Okay. For simplicity, I'm just going to trade this N. So they trade in the exact same spot, and they're going to swap. I'm going to tape them back together. In reality, each chromosome gets mixed around 2 to 3 times up and down from the top to the bottom. So you can see how crossing over can create a lot of genetic diversity. It's going to mix up the allele combinations that this father got from his parents so that what he passes on to his kids is not in the exact same combination. Okay, that's good enough. We'll leave this one as is. Okay, that's all in prophase 1. Metaphase 1, we line up homologous pairs across the middle. So again, 1 and 1 was a homologous pair, so they meet up here. 2 and 2 is a homologous pair, they will meet here. And the X and the Y, again, they kind of go together as a set, as a sex chromosome, they will meet. How I lined up this metaphase 1 is independent assortment. I could do it this way, right? I could trade. Chromosome number 1s are not talking to chromosome number 2s, are not talking to the sex chromosomes. They are only talking to their other homologous partner. Are you on one side and I'm on the other? Okay, we're good. So this is how siblings become different too. Like the siblings that result from over here, like the sperm, it's gonna get a lot of the grandfather stuff, right? And the one on this side is getting a lot of the grandmother stuff. I don't really wanna do that, so I'm gonna kind of shuffle this way. But it is totally random. We are working with just 3 homologous pairs. In human sex cells, you got 23 pairs, so it gets a lot more complicated. But that creates a lot more randomness and diversity. So that's your metaphase 1. Anaphase 1, the homologous pairs say goodbye to each other, and these duplicated single chromosomes are going to move to either side. This is what you're going to be drawing into these first 2 circles. Okay? So in this circle, I have a duplicated chromosome 1, I have a duplicated chromosome 2, and I have a Y chromosome. Over here, duplicated chromosome 1, chromosome 2, and an X chromosome. So the sperm that results from here will be forming male children because they're donating Ys to the mother's X. The sperm that gets fertilized and goes to the egg here will be forming females because they're going to end up with an X from their dad and an X from their mom because women can only donate X's. At least for humans. Some lizards and other animals change it up. Okay, that's anaphase 1. Telophase 1 is just these like become whole cells. So that's technically like what this part of the drawing is. So now, prophase 2. We basically have spindle fibers reforming and DNA kind of getting moved around. There's not a really dramatic change in prophase 2, but metaphase 2, we have 2 cells now that are lining up everything in the middle again. Okay, and how that happens still could like rotate and flip around a little bit, right? So lots of randomization and genetic diversity occurring with crossing over, independent assortments, and don't forget even mutation back in DNA replication. You could have made mistakes and these may not be exact matches anymore because of a mutation. Okay, so this is metaphase, 2, because we got 2 cells going on. And then anaphase 2, we break our sister chromatids apart. This is where if you had the masking tape, they're actually separating. 1, 2, it's just one cell, another cell, separating, separating, separating. So each cell, I'm gonna like draw lines, has a chromosome 1, each cell has a chromosome 2, and each cell has a sex chromosome. For males, half of them will have X's, half of them will have Ys. These are the 4 sperm that get formed in this one round of meiosis. So that's what gets written in across the bottom. So I'm gonna actually group them up so it just kind of makes a little more sense to me because chromosomes aren't all straight in a line. Cluster up in a nucleus. Okay, can't mess this up. This goes there, this goes there. Okay, so each one of these is a sperm. All of these sperm we would call haploid because they only have one of each type of chromosome, a 1, a 2, and an X. A 1, a 2, and an X, a 1, a 2, and a Y, 1, a 2, and a Y, right? If they were diploid, they would have looked like the first cell where there's 2 of chromosome 1s, 2 of chromosome 2s, 2 sex chromosomes. For a male, it would be X and Y. But this is our ending result. So these are your 4 cells that will go down here. And then as we roll dice to make children, this would be like a dice roll 1 and 2, a dice roll 3 and 4, 5 and 6, 7 and 8 for our 8-sided die. We can't split up 4 things with a 6-sided die, so we've got the special ones. Let's hypothetically say that it's this one. I'll keep these gathered together, and we can just use another one. And let's say that I got these donated from the mom. Okay, so this is egg, this is sperm. Fertilization, they come together. Chromosome 1, chromosome 2, and an X chromosome. So this fertilization event is going to recombine a haploid cell and a haploid cell to make one diploid cell. So I'm going to erase, let you kind of see that. 1 and 1, 2 and 2, X and X. It's going to all combine into one cell. This is the first cell that becomes the embryo or the zygote, like for the next generation. This is their first child. So I'm going to group the homologous ones together just so that I can see them. So I'm putting the number 1s together, the number 2s together, and looks like this is a girl. We have 2 X's, okay? So we have our homologous pairs back. This is a diploid cell again. Okay, 3 homologous pairs, or 1, 2, 3, 4, 5, 6 chromosomes. For humans, this is the difference between the 23 chromosomes in the egg and the sperm and the 46 that are in a complete cell that leads to a person. So lots of mitosis occurs. You make more and more and more cells. But what does this child look like? Their overall genotype, they got a big E and a little e, 2 big N's. Oops. They got a green and a red for body color, and X's that have capital F's. So this character is fuzzy. This is brown. Pink nose and eyes that pop out on the top of their head. So exo eyes are kind of outside of the body, kind of stuck to the top of the head. I can't do a pink nose, but they're brown and fuzzy. And they're a girl, so eyelashes, I guess. And if you make another child, it's just another grouping of the parent's gametes. So I'll get rid of this kiddo. I mean, just not get rid of, but, you know, move aside. And I'll kind of grab another set. So one of these, one of these, one of these. These are coming from the mom, and I'm going to grab a set of the sperm from the dad, just random. You will be rolling dice for this. Okay, so this child, we've got little e, so that's eyes inside the face. Big N, little n, a pink nose, big N is dominant. B-O for color makes pink, blue and orange, as close as I could get. And this one's a boy, and he's got only a lowercase f, so he has the recessive trait of being bald. So smooth body, eyes that pop out of his head, hair just on the top of his head, pink nose, and I'm going to give him like a shirt because he's naked. And there we go. There's my boy. Okay. Watch the video again if it helps. You are welcome to come in after school, before school. I got first lunch this year, and I have 5th period prep. If you don't have anything going on in 5th period, you could come on down and practice again. Okay, thanks.